Hello, my name is Dr. Kirsty McLaughlin and I work in the Agri-Food and Biosciences Institute in Northern Ireland, and I'm here to give a talk about the potential use of eDNA as a tool for surveillance in forests. First of all, I'd like to give just a little bit of background of AFBI, who we are. So the Agri-Food and Bioscience Institute is a leading provider of scientific research and services to government, non-governmental organizations, and also commercial organizations. We are sponsored by our key stakeholder, which is the Department of Agriculture, Environment and Rural Affairs, or DERA. Our customers include a range of local, national, and international commercial companies, Northern Ireland and UK government departments and agencies, and associated bodies in the European Union. What we do at AFBI is we undertake basic, strategic, and applied multidisciplinary research for DERA and other government departments and agencies, also industry and commercial organizations and bodies representing the agri-food industry. We provide evidence-based scientific advice to government policymakers, regulators, and industry on environment, agriculture, and food issues. We provide a wide range of statutory, regulatory, and commercial analytical services such as chemical and microbiological analyses of food and animal feed. So AFBI contains a number of different divisions, and within that there are different branches and labs departments within those as well. So where I am based is in the plant health department. In that we have a series of labs. We have crop pathology, which is looking at the pests and pathogens of crops, forest pathology, which conduct forest surveillance work, primarily for fungal and bacterial infections in forests. We've got the entomology department, which look at forest surveillance of pests. We've got nematology that look at the nematodes that affect primarily crop species, and then myself, which is within the molecular biology group, which provide molecular services to the other departments mentioned. And also, this is the department that carried out the research that we're going to be discussing today. So, to introduce the work, so plant pests, including pathogens, threaten forests. They reduce carbon sequestration, they disrupt trade, and are costly to manage. The Food and Agricultural Organization estimates that up to 40% of global crop production is lost to plant diseases each year, costing over $220 billion. The International Plant Protection Convention has adopted a series of international standards for phytosanitary measures to help national plant protection organizations, or NPPOs, to protect plant health. These plant protection organizations are responsible for implementing phytosanitary measures that aim to prevent the entry of plant pests into a territory and also control plant pests when they are found. The plant protection organizations are also responsible for conducting national surveys to demonstrate that their jurisdiction is free from regulated or emerging pests. Any control actions taken by the plant protection organizations must be balanced based on the serious socioeconomic impacts of establishing phytosanitary controls relative to the potential impact of establishment of the pest. So traditionally, forest surveys involve visual inspection of trees for symptoms of disease. However, this process is time consuming, and by the time symptoms are observed, often the disease may already be widespread. Once the disease is already established, eradication can be costly. In the UK, the cost of eradicating the Asian longhorned beetle in southern England in 2012 was estimated at £2 million. However, eradication costs can be dwarfed by the recurring management costs if a pest does become established. For example, management of Xylella fastidiosa, which is a bacteria, their outbreaks in Europe is projected to cost over €20 billion. And ash dieback, which is present throughout the UK, is predicted to cost the UK £15 billion over the next few decades. Eradication schemes often fail because the infestation is too widespread. Conversely, eradication schemes are more likely to be successful when they're implemented early in an outbreak. Therefore, we need new methods of surveillance to help plant health authorities monitor and protect forests from disease, especially methods that can potentially detect infestation before it becomes established and widespread. Previous research has shown that metabarcoding, which is identifying multiple species within a single sample of environmental DNA or eDNA, can be used to identify plant pests. The aim of this study was to assess the efficacy of using eDNA from rainwater samples collected in forests as a new surveillance technique for plant health. So this study collected eDNA from rainwater that had passed through the canopy of oak, pine, spruce and ash trees, and also rainwater trap that was placed in an open field in 5 forest sites in Northern Ireland. So rainwater samples were collected monthly for 1 year, from October 2022 to September 23. There were 2 oak, 2 pine and 2 spruce replicates and 1 ash and 1 non-tree sample. Rainwater traps consisted of 2.5 litre black plastic containers, as you can see demonstrated on the screen, with a funnel attached, and these were placed under the tree canopy on a wooden support structure that was 1 metre from the ground and approximately 1 to 3 metres from the trunk, depending on the canopy spread. Funnels had mesh placed over them to prevent debris from clogging the funnel, and the non-tree field trap was placed in an open area in a field in the forest with at least 15 metres away from hedges and trees. The 2.5 litre containers were collected at the end of each calendar month and replaced with sterilised containers, and the collected water samples were returned to the lab on the same day and stored at 4 degrees, i.e. in the fridge, until filtration could take place. Forest sites were selected to transect east to west across Northern Ireland, as you can see from the map. The west of Northern Ireland generally experiences cooler temperatures with higher rainfall than the east. Now, the distance between the easternmost site, which is Mount Stewart, and the westernmost site, which is Loch Navar, is approximately 150 kilometres. The forest sites in the study served a range of purposes. For example, Loch Navar and Dava forests are dominated by densely packed monoculture stands of spruce and pine, which are used in the production of timber. By contrast, Mount Stewart, Loch Gall and Hillsborough are smaller forests with a broader range of tree types and also include recreational areas like lakes, trails and play areas. So just to give an overview of the process, a maximum of 450 mils of water was filtered via vacuum filtration through a 0.2 micron filter. This was then stored until eventually extracted using a Kyogen Power Water extraction kit. Elutions were then sent to an external company for amplicon-based short-read sequencing using the Illumina platform. We used the region 1 of the internal transcribed spacer gene to detect fungi and oomycetes present in the samples. So of the 480 planned samples that could have been collected, 28 could not be collected either because a trap couldn't be placed or it was toppled over or there was insufficient rainfall in the sample. So the study generated 9,124 ASVs, so that's assigned sequence variants, so groups of similar sequences together. And this was done using a trained classifier, which was built on the latest Unite database, which is the database used for fungi. A total of 4,206 of those, which is about 46%, could be identified down to species level. 4% or 312 of the sequencing variants were unassigned. So these were ones that could not be identified either as fungi or as anything. Sequence variants that resolved down to species level were compared to known fungal and oomycete plant pests identified by the governments of Australia, Canada, European Union, United States of America, United Kingdom, and the European and North American plant protection organizations. After this filtering process, there were 179 sequence variants, which corresponded to 65 plant pests, 63 fungal pests, and 2 oomycete pests. 9 of these pests were present on the UK Plant Health Risk Register, and it's these 65 pests that were taken forward for statistical analysis within the study. So the metrics that were used, just to define a few things. So detection was literally the occurrence of any of the 65 plant pests within a sample. Diversity was the number of unique fungal and oomycete pests. We used generalized linear mixed effect models to assess the effect of site, tree trap, month and season, and we also used ANCOM BCs, which are the analysis composition of microbiomes with bias correction, which are used to visualize differences in community composition between sites. So it's a way of visualizing if there are more or less than specific species within your community. So some of the main findings. So the month with the greatest number of pest detections was November, as you can see. So detections are the blue graphs on the left-hand side of the screen. The lowest number of detections was in May. Seasonally, autumn had the highest number of detections of pests, while spring had the lowest number of detections. This isn't necessarily unexpected, as there are a lot of fungal spores present in the autumn. Most of the pest detections occurred in traps placed under the canopy of pine trees. However, as I previously said, there were twice as many rainwater traps placed under pine, spruce, and oak as there were under the non-field and the ash trap. So when the number of replicates is controlled for, it's actually the non-tree field trap that had the highest average number of detections. The site with the largest number of pest detections was Loch Navar, whilst the site with the lowest number of detections was Dava. So if we look at the, so that's trends in the data. So if we look at the GLMM results for actual significance, the month that had month had a highly significant effect on pest detection, with most detections observed in November. Tree trap also had a highly significant effect on pest detection, as did season, with the non-tree field trap and autumn having the highest number of detections. Pest detection was not significantly affected by site. Next, if we move on to pest diversity, so that's the number of unique fungal and oomycete pests found in a sample. So those are the green figures on the right-hand side of the screen. So pest diversity, first of all, let's think of the trends. Pest diversity was highest in July and in September, whilst the month with the lowest diversity of pests was May. So seasonally, it was summer and autumn that had the highest number of unique pests, with the lowest number of unique pests found in spring. The tree trap with the highest number of unique pests was again the non-field tree trap. This was again despite being roughly half the number of the other tree taps, because remember we had one replicate of non-tree trap and ash and 2 pine, oak and spruce. The highest number of unique pests was in Loch Gall, followed closely by Mount Stewart. Dava had the lowest diversity of pests, followed by Loch Navar. So again, if we look at the effects or the results of the GLMM, diversity significantly differed across tree traps, with the most diversity seen in the non-field rainwater trap. Diversity was also significantly affected by month and season, with July and autumn showing the highest diversity of fungal and oomycete pests. And finally, again, as with detections, fungal and oomycete diversity was not significantly different across sites. Moving on to the findings of the ANCOM BC, so that's analysis of compositions of microbiomes with BIOS correction. This was carried out on 4 different forest sites. This is the graph on the right-hand side of the screen. Using DAVA as the reference site. Each graph represents one of these 4 sites, showing an increase or decrease in statistically significant log-fold changes in fungal and oomycetes pests relevant to their abundance in the reference site in DAVA. Increases in the log-fold change are blue and are labeled as enriched, and decreases are in orange and are labeled as depleted. The ANCOM BC was also carried out on the 4 different trap types. So that's the left-hand side graph with using spruce as the reference tree trap. And you can see the names of some of the species that were significantly enriched or depleted in those areas in relation to the reference. So for example, if we look at Mount Stewart, Alternaria infectoria was significantly enriched in Mount Stewart compared to in Dava, as it was in Loughgall and also Hillsborough. What this can potentially do is help to target traditional forest surveillance techniques by showing you where you are potentially more likely to find certain species if these are the species that you're looking for. So for example, Alternaria infectoria is significantly enriched in Mount Stewart, Loughgall and Hillsborough in comparison in Hillsborough in comparison to Dava. So those are the 3 sites that you could potentially go to look for evidence of that species rather than going to Dava or to Loch Navar. So of the 65 fungal pests found, 9 of them were on the UK Plant Health Risk Register, but none of them were actually quarantine pests. So we didn't find anything that was labelled as a regulated quarantine pest. We did find 2 pests in our data that had not previously been found in Northern Ireland, which shows the possibility of finding such things using next generation sequencing and using eDNA. So the findings in the that I've discussed thus far, it shows that these data could be used to help target traditional surveillance techniques by showing perhaps when and where are the best forest sites to surveil for specific pests. It also shows that we are capable using this technology to find plant pests and to potentially find plant pests that haven't previously been found in Northern Ireland. But as with everything, we have caveats and there are questions that still need to be answered, and we'll have a look at those now. So just to finish off, last couple of slides here, expert judgment is critical to interpreting the output of next generation sequencing data, NGS data. The information produced by sequencing needs to be examined in tandem with the known biology of the pathogen, the relevant phytosanitary legislation, and the scenario from which the samples were connected, and also the wider taxonomic status of the pathogen and its close relatives. Next generation sequencing is still a relatively new method in plant health diagnostics and surveillance, and a period of time will obviously be needed before limitations of the method can be fully understood and addressed. In particular, the uncertainty of taxonomic assignment needs to be clearly communicated to policymakers. A secondary confirmation of any plant pests found through sequencing will be required before implementing any phytosanitary controls. A way to show some of the caveats and some of the limitations of this kind of technology done in this way thus far are to look at or to answer the question, did we find what we expected to find? Well, we did not find two important species that are known to be present in Northern Ireland forests. So we did not find Hymenoscyphus fraxinus, which is the causal agent of the ash dieback, and we did not find Phytophthora ramorum. And I just want to very briefly go over some of the reasons as to why. So for some pests, aerial dispersal is not considered the main way that they spread, and thus rainwater trapping might not be suitable for their detection. However, it is noted that we were able to find a couple of species of Armorillia, which are soil-borne fungal species, but we were still able to connect those. Maybe we need to collect eDNA from soil and leaf litter and other water courses to be able to pick up a wider range of species. Secondly, the primers used in this study may not amplify some of the pathogens that we know to be present. So using a different primer set or using multiple primer sets might help use a greater range of taxonomic marker genes to detect a broader range of pathogens. For example, some of the studies that have used eDNA to find Phytophthora have used specific Phytophthora primer sets in order to do that. The missing pathogens, it's always possible that they're not present in sufficiently high concentrations to be detected in our collection and filtering approach. However, we think that that is pretty unlikely given how widespread ash dieback actually is. Short read sequencing technology often doesn't provide sufficient resolution to differentiate between species of the same genus. So while we failed to detect ash dieback, another study that's currently unpublished that used long read sequencing technology was able to find it. So, it is possible to find it. It may be because our technology was short read. Also, species identification using the ITS region may not always be suitable for precise species level resolution. Now, the ITS region is the official barcode for fungal species identification. It might not be effective across all fungal taxa, and maybe it would be better if we use multiple taxonomic markers to improve species resolution. So just some take-home messages. So this study illustrates that the potential use of eDNA in plant health surveillance. To our knowledge, this is the first wide-scale application of eDNA from rainwater to detect fungal and mosquito pests of plants present in forest environments. Going forward, the application of next-generation sequencing to detect bacterial and insect pests from forests and eDNA will be important in order to cover the wide range of pests that can threaten trees. Environmental DNA can provide authorities with an overview of what pests might be present in their forests and where and when these pests might be found, which should allow the plant protection organizations to focus their resources to those pests which pose the greatest threat. This approach that we used has the potential to enhance plant health surveillance efforts through early detection, shortening the time it takes to implement statutory action, which of course reduces the cost of control measures and can improve eradication and containment outcomes. So finally, I just want to say A thank you to my team and also the Agri-Food Biosciences Institute through which this work was conducted, and our funders, which was the Department of Agriculture, Environment and Rural Affairs, or DERA. More information about this research can be found in the recent publication in the Forests Journal. And of course, if anybody wants any more information on that or to discuss anything else, my contact information is provided. Thank you very much for your attention.